All right, so here we are, chapter number 5, amino acids, peptides, and proteins. This is going to be our entry into this major macromolecule of proteins. We are going to use this as our segue into jumping into chapter number 6, where we're talking about the structures of proteins, what proteins look like whenever they're actually functional and everything like that. But let's go ahead and get started. So, an overview of protein synthesis. This is also the central dogma of molecular biology, also known sometimes as the central dogma of biochemistry, and that is DNA is transcribed into RNA, which is translated into a protein. The last little element of that is, well, what follows that protein is that it gets folded into this unique shape. So if you'd like, you can envision this in a eukaryotic cell, in that you've got a messenger RNA, which goes to ribosome, and that at the ribosome, that messenger RNA molecule is translated into a protein. So that takes place, that transcription and translation is going to take place outside the nucleus. Okay, so whenever it comes to protein synthesis and proteins, there are 20 proteinogenic amino acids that you need to be familiar with. I am going to give you the structures. I will not give you the single letter or 3-letter codes of these or the names associated with those molecules, but I'll post that up on our Canvas page. Amino acids are covalently linked in a linear sequence, and that sequence these amino acids are going to assemble, and if they make a length, or if they make a polymer that's greater than 50 subunits, then it is a polypeptide or a protein. Under 50 subunits, then it's just referred to as a polypeptide or a peptide. So here's what a covalently linked amino acid chain is commonly represented as. So here's our linkages between each of the individual monomers. Now these have single letter codes because each amino acid has a single letter code. There are a total of 20 common protein-genic amino acids, and heck, there's 26 letters in the English alphabet, so works out pretty well. We get to save ourselves from having to remember 6 of those troublesome letters. Okay, so what are amino acids? They're building blocks of proteins. They function as individual units in metabolic processes as well. Now, imagine that you see this little I don't know, bracelet or necklace type thing down here. Well, each one of these different colored beads is representative of a uniquely colored, or a unique amino acid. So proteins are going to have different amino acid sequences. So there might be one that has 15 of the exact same amino acids, and that protein is going to do function X, whereas a different functioning protein is going to have a different sequence of amino acids. Now, what is an amino acid kind of in a nutshell? Well, I'm going to make one change to this model that we've got right here. I'm going to put one more H here, and I'm going to put a positive charge there. Okay, so an amino acid is a compound that contains an amino group, which is indicated right here, and a carboxylic acid group right down here. An alpha amino acid is, well, what we're talking about when we talk about our protein-genic amino acids, because they have this central carbon that groups or kind of links those two amino and carboxyl groups. And this is known as your alpha carbon right there in the middle. So if you think about the term amino acid, well, there's something that kind of, most people are sort of triggered by that, and they say, oh, I know exactly what an amino acid is. They're talking about the protein-genic amino acids. In the event that, but technically an amino acid is just a molecule that contains an amino group and a carboxyl group. So we use this additional piece of nomenclature, this alpha amino acid, to indicate that we've got this alpha carbon between those two functional groups. Okay, so our amino acids are amphoteric molecules in the sense that they act as an acid and a base. So here's our acid, or sorry, here's our basic side, and here's our acidic side. This is going to donate a proton at a low pH, and this other side will donate a proton at a higher pH. So these can technically act as buffers in two regions because they have pKa values for their carboxylic acid at about 2 and their amino group at about 10. Now, acidic and basic amino acids have a third pKa corresponding to their specific R group. Now, our general structure is like this. We already talked about that alpha carbon, that central atom, and then we've got our amino group, and we've got our carboxylate. We have a single hydrogen coming off of our central carbon, and then we have that unique R group or side chain that distinguishes every amino acid from one another. Now, amino acids can be classified into a number of different groups based on their interactions with water and based on kind of their functionality as well. Now, in terms of learning the structures and becoming familiar with them, there's tons and tons of different mnemonics, and some people might look at them and say, okay, well, what I'm first going to do is I'm going to say which of them have sulfur, for instance, and then which of the R groups have a nitrogen in them. There's tons of different ways that you can learn them, but the most well-accepted classification is based on whether or not, or based on their interactions with water. So those four distinguished classes are neutral nonpolar amino acids, neutral polar amino acids, acidic, and basic. So our acidic amino acids have a negative charge at a physiological pH, and our basic amino acids have a positive charge at a physiological pH. Our neutral amino acids are hydrophilic, but they are still capable of hydrogen bonding. That's not a but, they are. So they are hydrophilic, they're capable of hydrogen bonding. Our neutral nonpolar residues are hydrophobic, and they have aliphatic or aromatic chains. All right, well, I'll see you in the next part of Chapter 5.